So for those of you who don't know me, welcome. I'm Frank Simons, Associate Dean of Research in the college. It's a service role. I always like to remind people. It does have limited time limits, so, you know, when that clock rolls by, who show of hands, faculty here? You're in the pipeline, okay. 2 years, Jed. So we try to put together, over the course of an academic year, 2 to 4 college-wide talks that we think are relevant to research and its conduct that would be useful, of interest to our faculty and research staff. And for those of you who don't know, this week is for the University Research Ethics Week. And so we try to put 2 of those things together. And in doing so, I thought of Rebecca Davies, who agreed to come and speak with us. I'm going to do a very short introduction and make a couple of personal reflections on why this idea led Ellen and I to go over and meet with Rebecca and convince her to come and give a talk to the college. I'm going to first write up, I've already said it to her privately but publicly, you know, we usually get this kind of numbers out. We had good RSVP, and then, you know, we're starting to take names. We can publicly shame them later. So, but the way to think about this group is if they're here, they're interested. So that's probably a better way to go. But feel free to move about the cabin as you need to engage with folks.
Thank you.
And I'm going to point out, I'm carrying my yellow pad, which means nothing to most people. But the yellow pad for me is Frank being Frank the researcher. If I have a white pad, it's all administrative crap. So I'm taking notes and paying attention. So how do we get to Rebecca? Dr. Davies, who is a faculty in our College of Vet Med and an expert in our own backyard. So as part of being the Associate Dean of Research, I get the privilege, I put double quotes on that, once a month to sit centrally in the college. No, the Council for Research Associate Deans, CRAD, meets monthly. And there's always a variety of speakers with important information that is pushed out to all of the research associate deans and related folk, including Ellen. About almost 2 years ago, I think, Rebecca came and gave a talk on quality assurance, QA. And I remembered it very well. So when Research Ethics Week came up and the idea was to have a talk that tied into that, in talking with Ellen, I mentioned Rebecca's name. Why did her talk stick out to me? Because I had an emotional, visceral reaction. And if you read the Nature article that Ellen circulated, in the opening few sentences, it's something about almost throwing up.
That was my visceral reaction.
That was her visceral reaction when she got going on QA. So when Dr. Davies was giving her talk at CRAD, the opening few slides, I'm not proud of it, but my reaction, one, I was paying attention, which is not often the case in a CRAD meeting, but enough that I was paying enough attention that I was having kind of an emotional reaction of, what are you saying, lady? I think we have enough processes built into the scientific production pipeline, i.e. peer review, et cetera, that we don't need another layering of checks and balances. And so I started quite negative, and then the way I described it to her the last time we were talking, it was sort of an emotional roller coaster ride, again, which is not typical of a CRAD meeting, where I kind of rode the wave, and by the end of the meeting, I was like, my God, I felt like I'd seen the light. She's right, and this is important to mention that we tend not to talk about, particularly broadly maybe in the behavioral, social, educational sciences. And so, with that, we were able to convince her to come and give a talk, and I believe how you think about us may be, you know, the social sciences. So this is, I think, your first crack at putting out what you're thinking about in terms of quality assurance in front of, let's say, non-wet lab type folk.
That's right.
How about that?
That's right. Thank
Okay.
you. Thank you so much.
I'll stop my rambling. The floor is yours.
Okay.
Usually, I mean, we've got this thing booked for 90 minutes. You're not going to fill 90.
No.
And I don't know if you want to entertain questions
I
as
do.
you go, so you can run it however you need to run it.
Okay.
All right?
Thank you so
Okay.
much.
Thank you, and thank you for coming over.
You bet. And I am so happy to be here, and I really want to just get through my remarks because I am very hungry to understand what your thoughts are about this type of content, some of these ideas from the social science perspective for a couple reasons, and I probably won't digress until I've done my work, but I am really looking forward to, and I hope you have time to stay for some conversation about these ideas and one, I'm going to present these ideas, but I'm not going to present solutions of how, if you like the ideas, how do we put them in play? How do we bring them into our training programs? And so that's the big elephant in the room for me, and I'd also like your insights there. So the title of my talk is, and this is really what drives my interest as a scientist in quality assurance, is because I see it as a really good tool, a strategic tool for us to stand up for the quality of our work, based on the fact that we're going to be able to generate a lot of credible evidence that the rest of the world understands, I think perhaps better than a CV or a grant history or a scientific reputation. So I like the idea of quality assurance because it's what I think scientists can use to stand up for and demonstrate the quality of their work, which probably there has never been a better time to be thinking about that than the current environment. So I'm over at the College of Veterinary Medicine. I'm working in a program I call Quality Central, where I've stepped away from my endocrinology work, which is what brought me to the vet school. I was faculty advisor for the comparative endocrinology laboratory in the diagnostic lab. Did that work for many years, and I don't know if you had a chance to read the Nature commentary, but I was asked to help the diagnostic lab, which is about 15 different laboratory sections, meet an accreditation standard that we were probably very unprepared for, and that I knew nothing about. As scientists, I think we learn a lot about quality control, but in my experience, I had no training in quality assurance, and I talk about this topic pretty much all around the country and internationally, and I've yet to come up against or in front of a group of academics who were presented with quality assurance training either. So I really do think it's an interesting gap in our training program. So what I'm interested in is, what are institutions and what are we as scientists doing to respond to some of these questions and concerns about research rigor? Where's our voice? And you're going to hear me say this as we go on, that we really are rapidly responding to our stakeholders, the people who fund us, the people who publish us, but I'm not sure that we're driving that conversation as we might want to be. So I'm very interested in what are we doing to drive and establish some of those parameters or infrastructure about the way we do research, and be the ones to say these are what we think are best practices, rather than reacting to what other folks might think are best practices. And then if we want to take ownership of that conversation, what are the strategies so that we really can own that and use it to train the next generation? So I'm only going to spend 2 slides sort of putting up these pictures that I suspect many of you have seen before, right? About the current, and by current I mean anywhere from John Ioannidis' first paper there in 2005, and he, by the way, is a speaker this week, and it'll be really worth your time to go to that symposium. He's at Stanford. He's really been the voice of research on research and metrics for scientific practices, and showing us a lot of information that maybe doesn't help us sleep that well as scientists. So these kind of studies, there are many of them. I know there are certainly several in the social science area, certainly psychology, some in economics, some in computer science, and I'd love to hear from your perspective of others. Where we really aren't meeting our own personal expectations as scientists for our ability to replicate and gain from our work. This paper you may have seen as well. This is Leonard Friedman. He's an economist. He did this analysis of of multiple publications in preclinical type research. So again, many of my examples are going to be from the biological sciences area. So forgive me for that. And again, I'm interested in what you may have seen in your own discipline. But this paper gets quoted over and over and over again because he actually put numbers to the problem, and publishes here that based on their analysis of, I think it was over 700 different published studies, and their effort to try to say, if we wanted to replicate these studies, could we, based on what's provided in that manuscript? And their findings were that no, half the time they probably couldn't. And so if you couldn't reproduce it, they're saying that's research wastage. And according to their financial analysis, that's half of the United States full budget, and that's academic, contract research, industry research, is considered research wastage. And the reasons, the buckets of problems that he and his co-authors identified are what you see on this diagram. And if you're familiar with where the NIH is going now with some of their requirements for our how we present our grant proposals, they're asking us to do a little bit better in all 4 of those categories. So this has been very impactful research. So what I hope to do today is introduce this idea of quality assurance, because in my experience, we're not necessarily familiar with how this could be a tool for us in our own research environment. And then discuss it a little bit as a scientist-driven strategy, and I focus on that because the minute it's taken out of our hands, it may become something that's not nearly as useful. Because for us, if we want to put some infrastructure and consistency around how we do research, we want flexibility built in there. It's so critical to us. So I want to be able to present this as perhaps, as Frank mentioned, our initial reaction might be no way, but when we think about it from the point of view, if it's something we can design and we base it on our risks, it might be useful. And then provide some indicators that it might actually help. There are data out there that demonstrate that this might be something that could be useful. And so I want to show you a little bit about that. And then what I'm looking most forward to is this opportunity to brainstorm about this idea. What makes it, what are the barriers? What makes it sound good? What makes it sound terrible? What could you imagine in your environment doing either at a small level or a large level? And what should we be expecting our institutions to be doing to support us? Because I think that's the key. Okay, so whatever kind of science we do, and I took some of this from the websites that I went to do a little looking to see the kind of work you do, and I really was just amazed at all the different centers and strong research programs and strong research support that is in this program. And so it seems like amazingly fertile ground for research structure, research improvement. And I really like the carry 4 icons when I see research assessment, data and analytics, and program evaluation and innovation. It's just like, that's what we all want. That's what we're all trying to do. And one of the things about quality assurance is it gives you this opportunity to evaluate your own processes. Now, I know you're talking about evaluating educational programs, but just the fact that you've recognized evaluation as a big important feature, and yet as individual researchers, we don't tend to do that very much, right?
Sort of like a gratuitous picture of a bench in there.
I'll work on that. Continuous improvement. It's what we're all about. So no matter what we do, these ideas that I'm going to be presenting to you truly adapt to any kind of data. And so when we think about research, really there are those 2 aspects. And this is simple. This isn't rocket science, but this is what makes rocket science work, right? It's what we do. So that's the sound science, how do our research premise, our ideas, that critical scientific thinking, and then perhaps the more mundane, how we do it. What are we doing at that bench out in the field as we're collecting data from our surveys? And so I think as we look at those 2 things, I'm going to be suggesting that we're maybe targeting sound science with most of our interventions and perhaps neglecting research practices. This table comes from the WHO document Quality Practices in Biomedical Research. It's freely available online, published in around 2006, gives you a perfect blueprint for how to integrate quality assurance best practices in your research. And very few of us know about it or certainly have integrated it, but it's a wonderful resource, and I think this table so simply presents the value of thinking of these 2 different compartments. And I have sound scientific principles circled because I think there's a lot of evidence that this is where we're focusing. We are responding pretty quickly. The NIH is responding. The publishers are responding. Our research training programs, when I look at the support that you folks have on your website, places to go for when before your grant and after your grant, and study design and understanding bias. We're working hard in these areas. And now that the NIH has really asked us to work hard, there's just a very robust response here at the university to what training programs do we need? How do we meet this requirement? What words should we say? So there's a tension there. And our stakeholders, as I mentioned, the journals have, of course, they're a really important stakeholder, and it's not to their advantage when retractions occur, when work can't be reproduced. And so they have very quickly come to the table too. They've formed consensus group, they've created checklists, they've reoriented materials and methods requirements, and they're working hard to convince scientists that impact factor is a pretty poor measure of quality. So the journalists are helping, spread that message. They're also engaged in conversations about the incentive system. And so our stakeholders are working hard. NIH and NSF also has got new suggestions about how to deal with rigor. And a lot of the societies, the scientific societies, are also coming together, forming working groups to try to see how to support their members better with these questions. So this is just an example of the NIH new guidelines and website on rigor. So when we think about problems with research reproducibility, and I tend to like to get away from that word reproducibility, because wow, if we really wait to figure out if our work is really reproducible, it could take us a long time to answer that question. There are many reasons why we would expect some of our research not to be reproducible, but I think all of our work should be accountable, and so I like research accountability. And when I think about the threats to that accountability, we have what you see on this slide. The questionable research practices, that tends to be, that actually is a term in the literature. It's kind of a new bucket for what's wrong with how we do our work. And questionable research practices are, some of the hard things that you're making a wrong decision perhaps because it was a difficult decision to make. But these are the study design, the bias, some of the way we analyze data, the statistics we use, the tricks we use with statistics, our tendency to perhaps figure the statistics out after we've got our data rather than establish it before, our tendency to not bring those statisticians in early at the grant proposal stage. Some of those things are considered questionable research practices. And of course we have fraud, and I don't talk much about fraud. It's the kind of thing that it's only 2% probably of the problem with irreproducible work and retractions, but it's, the truth is for me with fraud, if we have people who are intent on fraud, sound science and good practices probably aren't going to deter them that much. Although I do think some of the QA practices would make it a little harder. They'd have to sign their names more often. They'd have to be, have more of a trail for them. But I won't talk much more about that. But then there's also just our routine habits, our total reliance, truly total reliance on research mentors, which is really interesting because when you think about how we feel about uncertainty in science and variables in science, and yet we tend to rely completely for our research training on variable mentors, and most of them do a great job. But if you think about the mentoring that goes on all around the country and all around the world, our uncertainty is a huge number, right? It's huge. So there's potential there for inconsistent training that could have an impact on our ability to do consistently good science. And so, again, this question of are we doing enough to address routine behaviors, the habits that we build, and how we mentor, and whether we're doing things somewhat consistently. And after having gone through my experience at the diagnostic lab where we integrated a quality assurance program, and I realized how much better we were afterwards, I mean, I was truly stunned by how much better our accountability was, our records were, and our ability to solve problems and find errors that we just wrote off as being, oh, it was a bad day. You know, so I think some of these best practices could help all of us. So now I'd like to step into, well, what am I talking about when we talk about quality assurance? And I think it's easier to think about it in terms of widgets and airplane parts and Toyotas, which is where it kind of got its start. So these are systematic approaches to work. Now I'm gonna say that and run with that idea, but I also want you to think at the end of this, if we all agree, you know what, it's too hard, we can't get a quality management system approach to basic research, well, there are ways that you could cherry pick and select some best practices that would help your work, okay? But the best approach, I think, is to use it as a quality management system for how basic research is done. And that's because the whole goal of these systems, and they're the same no matter if you're making an airplane part or research data, it's to improve and maintain the precision, so the consistency and accuracy, does it fit an expectation of a product? And also this to establish routine performance. And this is what usually gets our hackles up when we're, as scientists, because we're thinking we're not routine, we're special. We need creativity, we need flexibility, we need to, you know, change our ways based on what we're taking in and learning. But much of what we do is routine. And if we can make that consistent and have that established so that whoever we're training is going to do that consistent piece consistently, we're going to be a little bit closer to efficient and accurate work. So in our world where we're not making widgets, what we are generating are research data. The inference is another piece that, you know, is very important. And then the publications. We generate publications and they're very important to us. And then of course the next generation of scientists. So when we think of that product, now let's try to think about how quality assurance might work with that product. And as a systematic process, it's put in place and it generates evidence that the work is performed to a standard. So a whole part of a quality management system is you have to decide what your standard expectations or requirements are. And again, this is the beauty of doing this on a voluntary basis, because we as scientists can establish for our research lab, for our project, these are the requirements we want everyone to work under and meet, okay? So this concept really has become clear to me that as scientists, we don't really understand quality assurance, but our public absolutely does. They're very much used to having somebody else take a look at the quality of whatever it is they're considering buying or using and providing some data or feedback on that. And when I'm talking to people who ask me about this topic, and I say, well, we're just trying to put some standardized approaches around how research is done, the public, my friends who aren't scientists, they just are stunned to hear that we don't do this. We don't necessarily calibrate our equipment. We don't say that out loud, but we all know it's true, right? And this just really surprises people. So I think we have lots of examples of where a standard helps people feel that the product is more reliable and helps the people delivering it know that their product is more reliable. And trust and confidence is huge. It's huge for our biggest, most important stakeholder, which is the public, and whether they're going to care about science and trust science, they have to believe in what we do. And as I mentioned, we have best practices can apply to any kind of data. And this is what the system approach is going to look like. And again, this is probably what got Frank's heart rate up, is we start thinking about, oh my God, you're going to want, pardon me? Good to know, good to know. The reaction I often hear about is people who are familiar with regulated work and where you maybe have to have a standard operating procedure. The example is to open a door, to turn on a light, or to. And remember, that's not regulated work is different from what we do, but we do have. This is for me. I think about having an organized closet, which I don't have at home. But this is like an organized closet for our work. So basically it's this, you wrap your arms around the entire research life cycle. And you say, I'm going to put in some infrastructure, some expectations. We're going to write them down and communicate them and double check on them. And they're going to be associated with the equipment that we use. So we are going to have expectations that we've got data that show our equipment have been maintained and calibrated. Think about measurement uncertainty, and if you don't have calibrated equipment, even though your brain tells you, yeah, it looks good, it maybe isn't. And if you have to go back and troubleshoot through strange data, and you have no idea if that was one of those days, you've lost your ability to reconstruct your work. Same thing with methods, consistent approaches to methods. I'm sure that you would have some good examples in your world where two people doing something just a little bit different, maybe it's a survey, maybe it's an interview, can have a big impact on the actual measured results. So the whole idea here is what can we do to minimize our variables, address uncertainty, and make sure that whatever we're measuring has to do with the effect and not with the noise around our measurements. So personnel training, why do we care? It's just boring records. Truth is, again, it gives you that record when you go back and you've got some data that don't make sense. Was that person really trained in that, or was it a student who stepped in because someone was sick? Or can you demonstrate that people were competent and knew what they were doing and had the training, where they were entitled to the training and had they received it? Data, of course, this is huge. No one cares more about our data than we do. But how are we really developing data stewardship, data governance plans now so that we know where our data are? We know where they're being stored. We know whether we can read them 3 months from now or 3 years from now, or whether the titles are no longer descriptive. You all know what I mean. In the day of big data, things can get very messy in a hurry. So are we taking care of data? Consistent procedures can help with that. Error correction and monitoring this is the one that will probably be the last to be adopted in a research setting, but it's the one that impressed me the most in the service lab setting, of actually taking the time to look when things went wrong or when you didn't follow your procedures and find out why. It's been very illuminating. Usually it isn't because someone's a schmuck and they didn't do it right. It's because there was something missing in the step, or something that made it easy for that data to get disrupted. And then facilities and environment records. All of this attention to detail and attention to consistency where consistency is necessary generates these documents, generates the evidence that allows you to go back and reconstruct your data to source data, and also allows you to show that platform to someone else and say, sure, come on in, this is how we do things, let's reconstruct that data, let's find out why it's not reproducible. And the ability to have these records. And then, of course, when you have records, you can monitor, you can self-assess, you can evaluate whether the procedures are being followed. So as I've already said, it's very rare to find quality assurance in academic basic research settings. This is an interesting conversation now going on around the country and truly around the world. I'm going to a meeting next week in Germany where they have an organization called the IMI, Innovative Medicine Initiative. It's bigger than like our NIH. It's a conglomerate of funders to the tune of like $32 billion. And they put out topics and then they invite proposals to work on those topics. And one of the topics this year is the integration of quality management systems in basic research. One of the projects is funded by Volkswagen, go figure. See, these ideas always come after something's gone wrong, right? But Volkswagen is funding this project, and they're looking into, what might work, what's the best way, what might be the roadblocks. So, and that's just in the EU. In the US, I think there are conversations as well. So I think there will be changes in the future, but it'd be nice to be out in front of them. So since it's not in our routine, where are quality assurance systems used? And I saw some of you nod when I talked about working in regulated work. What you see on this slide, again, is translational medicine, biology type work where you're moving from basic research through new treatments, new drugs, new devices. And on the left side, we have basic research and discovery, which is unregulated because we need our flexibility and because it's not required to be regulated for safety and efficacy type requirements. Whereas in the development, preclinical development and manufacturing, we've now got people taking those drugs or using those devices. And so there's a real element there of safety, and we don't want to do this if it's not going to work. People are signing up for these studies. So in that case, it's code of federal law where you see GLP, which is good laboratory practices, good manufacturing practices, or good clinical practices. So that's by law, you need to do work with quality assurance systems in place. And that's because they're known to help safeguard the data and safeguard the participants, okay? Whereas where we work, we are pretty much allowed the creativity and flexibility we need. But when you think about this and you realize how the research ecosystem, all those folks over there trusting the device and the drug and the intervention, it's all built on the work we do. And it's those folks that we're now saying only 11% of the work can be reproduced. And these are studies on people in their last clinical trial. So it seems to me that this would be a wonderful place to have a voluntary commitment to a certain structure or a consensus-based standard so that we can make sure that they're standing on strong shoulders and that the influence of our data is going to the greater good. So if these systems really do generate credible evidence for research quality, and that's just another cartoon of what's involved with the system approach, well, if we're not doing it, if we're not training it, if we can't generate that evidence, are we really supporting our scientists and that precious data? And this is where I start nudging the institutions, because I think if we wait for every scientist to try to integrate a quality management system, it's hard, it takes a long time, it takes resources, and it's easy to slip back because it takes time and resources. And if you don't have the motivation or the push from your institution, it also becomes less of a priority, right? So I think we probably aren't supporting our scientists and their data as much as we should be. And this is an important question about our trainees. We all know that our trainees going out after their PhDs, they're not enough academia jobs for these folks. We need them to go out into other careers, and some of those careers require and expect the ability to understand working within a system approach to data. And I've had several people from industry tell me, and this is their words, that they hate hiring new PhDs because it takes them a year and a half to get them up to speed with working in a quality management system that they feel more resistant to standard approaches and expectations and monitoring and, putting up with some of the irritating aspects of a system approach, which, to be honest, it gets easier. Pretty soon you're not even noticing it. It's just part of how you do your work. So I'm hoping that's my argument for where maybe we're not doing as much as we should be doing. And then I'd like to just bring up, this is not a new idea. So this idea has been put forward since the 1990s, 2005. These resources are freely available that you see here, and if anyone's interested, I can send you a list of freely available resources that make very good cases for how to do this and for why to do this. This is the WHO document. And these others come from Research Quality Association in Britain, freely available manual that's really easy to take on board and put to work. I used this other quality research one I had a chance to work with some MD, PhD trainees last year, and we used that to try to integrate it into their project. And basically, these are the kind of questions they help you think about in terms of quality practices. When you think about your data, wherever it comes from, these are the attributes or characteristics of good data. Can you say that you could show and demonstrate that your data meet those requirements? How are we for documentation? Do we have records? Do we have any way to, other than trust and the fact that we showed someone how to do it, know that things are being done consistently? Do we have these training records so that we can go back and be sure people have had the help they've needed to get to know how to do the work? And this one, dare we even ask about care, maintenance, and calibration of equipment? And this is a big issue because I'm sure you folks know we can get the money through infrastructure equipment grants to buy a nice piece of equipment, but it rarely comes with more than that first year of service contract, right? And service contracts can be thousands of dollars a year. That's difficult. So this takes me back to the WHO document and this idea that perhaps we are running a little lean on good quality practices. And a question that I'd like you to think about now is, where are your quality checkpoints? As you think of the research you're doing or the research you're directing, at what points along that research life cycle from plan to completion are you getting the credibility and trust you need to feel like you can sleep well at night about how your data are? And this slide is just meant again to give us a little insight into the sound science. I really think we are working hard on that. Improvements are still needed, and there are new evidence for that being published every day, but we're working on it. We certainly have the research integrity piece, a long-standing effort in university or American universities to address fraud, plagiarism, budgets. But the help at the bench in the field may be less present. And as I know I've repeated that so many times, so I just wanted to give you a couple ideas of what am I really talking about between those two. And you'll see, obviously, that the good quality practices are the same buckets that a quality assurance program helps you address. And when we talk about research integrity, we're often thinking about honesty and subject protection and animal protection, but there's also the piece of the secure research record, the accurate and secure research record. That's data integrity as well. So do we have those records that we need throughout the whole cycle? This paper again is Leonard Freeman, who did the economics paper. He's part of the Global Biological Standards Institute. These folks have a great website. This is a white paper that they have written about looking at reproducibility from the last maybe 10 years going forward to 2020 with the interventions. And there are many suggestions of what we should be doing and what are being changed, and then where we'll be in 2020. It's a very nice summary of this whole field of reproducibility. And so when I looked at that, I thought, well, how's it going? Have we got this covered? Are we moving in the right direction? And this, I'm hoping that you can see this in the back. This is from that paper, and it describes what you're going to be very familiar with, the research life cycle. And then he's identified what he considers the normal quality checkpoints. And can you see in the back the green? Okay. So if we start up at the project idea, the brown checkpoints are ones that we all recognize. They're broadly used. Grant review, peer review, ethics, IRB type work. Then there are some that are moderately used, journal requirements, institutional review. And then in the gray are the rarely used. Okay, so perhaps after we have a experimental design, there's a PI review. Perhaps as we're working on the protocols, we've got a lab manager or PI review. A lot of the fraud cases actually come down to lack of sufficient review by the mentors and PIs. We've got PIs with so many trainees, and there's a lot of trust going on there. And so this is perhaps rarely used PI review is a little disconcerting. But as you look at that, if you look at this, and I think this is a good reflection of what our reality, this whole area from once the protocols are in place to when the data is analyzed, there's precious little going on there in terms of quality checkpoints. And that's where our data and metadata are. So everything that we care about is in this, at the bench, in the field with our surveys. Very few quality checkpoints going on there. And since we know that data and metadata is what's critical for reconstruction, we all think we're probably doing fine. I look back at my PhD and I wouldn't want to look too close at some of my data and metadata, but in general we feel like we've got this, right? However, if you look at some of the literature, some of this new research on research, which is fascinating, this is Melissa Anderson's work. She did a survey of NIH funded, which I think is important because that suggests the mentors are pretty strong. You're not going to get funded by the NIH if you're not a strong research team. Early career scientists, about over 3,000 of them, an anonymous survey, and these are some of the behaviors that were reported. You see the top three kind of hitting that sound science piece, right? But interesting to me is inadequate research design, sponsor pressure, cherry picking data, 10 to 15% of the sound science bucket. Nearly 30% say, and I would be right there with them, that they're not all that proud of their record keeping, their ability to reconstruct their data. This excites me because it's low-hanging fruit. This is so fixable. Some of these other issues are difficult. Study design can be difficult. Bias can be difficult. We can fix records. We really can. Here's another one. Again, these are primarily you guys can feel smug because this is primarily biology, that dirty laundry here. But this was an interesting paper. All they tried to do was go back into the literature, important studies using important reagents, and ask the question, could we repeat this study? And to repeat this study, we need to know what they used and its unique identification so we could figure out what it was and where to get it, right? We would expect that from our peers, yes? 54% of the resources were not uniquely identified in those publications. Okay, here's another one. This one has to do with mislabeling. It's a long story in a genomics paper where they looked at, and I'm not a genomics person, but based on the sex, you should be able to have a certain genomics pattern, okay? That's the basic principle here. They went back into these papers and said, based on the data reported, does the sex match the genomic data that they reported, all right? Simple test, 46% of the data sets had mislabeled samples, okay? One more, anybody who works with Excel knows you can get into trouble with Excel, yes? So they basically looked at looking at data sets with these default settings that, unbeknownst to you, change your data. 1/5 of the papers had gene conversions. So again, record keeping, not double checking, low-hanging fruit. So record keeping issues that we could help address. So to my mind, we really need some strategies to address some of these. Those are big numbers. Those are embarrassing problems, right? So I think, and they're fixable. It's very difficult to encounter problems that are just so complex that you don't know where to start. These aren't those kind of problems. And then I'm nearly done here. This paper, has anyone else had a chance to see this? This is a very interesting paper written by C. Glenn Bagley, who was also involved in the commentary on the cancer studies, where the Amgen study, where they found they could only reproduce 11% of these very important pivotal cancer studies. So he and his colleagues here, and Ulrich is the fellow who's organizing this meeting in Berlin next week with Volkswagen. So he is very engaged in the quality program now. And they're calling out, they are saying, and this is worth a read, they are saying institutions need to do more to address this situation. And when you look at this slide, I think it really sort of reflects the position of scientists in this environment. We have the funders who are trying to hold it together, but they've got ways to address this. They can address some of these issues through their requirements. We have the journals who are cracking, but they can also stick themselves back together. But we have this poor beleaguered scientist who's trying to work and get things done, trying to put all of this infrastructure in place around themselves, while the institutions are maybe a little bit asleep at the wheel, not moving fast enough to address research rigor concerns. So they recommended there's things that they call good institutional practices, and this is a summary of some of those. And as you look at those, as I look at these, I see a lot there that's just screams quality assurance to me. My husband says, when when you're a hammer, everything looks like a nail. So for me, when I see using established standards, do we need a standard of consensus standard of how we do work? Records, training associated with standards, incentive and evaluation systems, monitoring. This is a sea change for the way we do basic science. But then you read here that's interesting, and this is probably going to drive things in the next 5 to 10 years. So we're probably going to be with quality where we were with animal welfare and IACUC. We probably are, unless we quickly own this ourselves. So their expectation is we should be owning this ourselves. All right, and as you go back to this slide and think about those big problems, some of this low-hanging fruit could reduce those percentages probably significantly. This is the Nature commentary about maybe it's time, and in that article they interview several different scientists who have voluntarily put this in place, and they talk a little bit about the pain and the joy of that experience. So I'm interested in how this might sound to you folks when I look at what you do, and again this need for sound science and good practices and teaching. How do you feel about this? What is possible? And so now we're at that brainstorming part where I'm going to stop talking and say, I think we have to be quick, we have to be nimble and get in this conversation as scientists, because I'm not hearing a lot of that out in this conversation. And if we don't, we'll be led. And I already see a lot of evidence of that, because the people who are worrying about research rigor now in terms of the NIH requirements, what I'm hearing from them is, just tell me what to put on the paper. Well, that's discouraging. We should be saying, I want to put this on the paper because this is what I do and how I do it, and I can show you. Okay, I'll stop there.
Can you go back like maybe 4 slides?
Yes.
That one, GIF is the big title.
Yes.
So I'm going to.
Good institutional.
So
This one or this one, my favorite? Okay.
So right there, I sort of had my visceral reaction that I had when I first met
Yes.
you in the CRAG context where you started earlier with.
Yes.
And if I have to wear my institutional hat, which I will just.
Yes.
You are reacting to this, is, so the institution's back to the other, the visual metaphor, but they
Yeah.
aren't doing their part in this crackling, crumbling thing. And yet, my gut reaction is, so we gotta put in, we have an IRB.
I know.
And we hate it, and
Yes.
as a scientist, I hate it when they tinker. And
Yes.
our response is, don't mess with the science.
Yes.
Just on the ethics.
Yes.
And I'm just not sure where.
Yes.
Where this is supposed to fit. And I
Yep.
get the sentiment that
Yep.
if we don't take control
Right.
of our quality, someone will tell us how to take
Yes.
control of our quality.
So there's a couple answers to that. One of them is, in the whole world of quality assurance, you are not meant to mess with the science. I mean, there's meant to be a line there where you're meant to support and help them get their infrastructure in place and advise on mitigating risk, but you are not meant, the sound science piece is owned by the scientists. So these things are, in my view, when I talk about science-centered, strategic, and risk-based, the scientist needs the support maybe of perhaps it's QA people, or perhaps it's the right tools and templates to evaluate their risk and figure out what kind of record keeping might help them mitigate that risk, or what kind of procedure might help them mitigate that risk, or what kind of electronic tool, maybe an electronic notebook is going to help in this area. Basically, I think we need to figure out where the risks are and then find out what tools will help. And when I think about the institutional approach, I think maybe that means that we have, just like the library folks now are just doing wonderful things helping with data management plans, data curation. They're bringing expertise in that we all need, right? Well, maybe there should be some support of quality assurance folks who can advise researchers, or there could be a quality management system software that you could have access to, that you could put your documents in. That's kind of what I mean, not to establish those standards, but I think that's a really important question. Someone's going to establish those standards. It's already being talked about. There are societies who have said our standard for basic research should be similar to ISO 9001. Anyone? So that is where you have a product and you put down your requirements and you show that you satisfy your stakeholders. So in theory, it's a really good idea, and I actually really like that standard. But do we really want that as our standard? So I think from the institutions, they should be having this conversation. We should be driving. We can say what our standard is, and then we meet it. But no, I agree. Oof.
So are you imagining sort of a system in which, I'm thinking that one way to go would be for the institution to require researchers to indicate where on that continuum their research falls, preclinical or clinical, or where, and then that puts you into sort of like our current ethos system that would be the good clinical practices or the good lab practices, or and you have to insert your standard operating procedures accordingly.
Well, that's kind of what we have right now. So if you're on that blue frame, you have to meet those requirements. What I'm dreaming of is on the basic frame, where we think about our research and we maybe have something. I'm going to make this up in a very simple way. That we have on our website, or when we're training, we have a quality commitment that says, maybe we're going, for our routine procedures, we're going to have standard operating procedures, and I'm going to have a laboratory management SOP that tells all my trainees, this is where data goes, this is how we share it, this is how we do this particular method, and I'm going to train you to do it, and I'm going to train the next person to do it. So that's what I'm picturing is an understanding of a quality infrastructure and some way to implement that in your project based on your risks. If we think about it from the institutional level, that's too big of a dream even for me, where we'd say we have a quality assurance support program that actually does the monitoring. That's what's needed, because we all know monitoring helps, but we also, that's really tough. We couldn't, the scope would be way too big. That's a piece I didn't talk about much, is in real quality systems, monitoring is a big part of that. And I think it's really important. And I think that's going to be a piece of what's coming as well. But that monitoring can be self-assessment, it can be peer assessment, it can be checking lab notebooks as part of your lab notebook for, the kinds of things that are easy to go wrong. Not sure if that's helpful.
You're an epidemiologist by training, right?
Endocrinologist.
Oh, I was
Yeah.
I was gonna say, have you moved in, are you becoming like an interventionist? Have you tinkered
Oh.
with other systems, people in their world to say, put this in and
Yeah.
things get better?
That's what I would love to do. And that's what we did in our laboratory. And what I've tried to do we got an NIH supplement to tinker, to work with these 12, I had 12 trainees, MD, PhD students, and that to me that's the goal. Maybe it's all through training, where we worked with those 12 trainees, we said, let's take your project and let's just adapt SOPs, equipment records, test validation to your project so that you've got a nice clean closet and you know where your data are. And then try to maintain this as you go forward into your research career. Have expectations that everybody understands and knows about, have requirements for routine work, have an SOP on how you handle your lab notebook, your creative piece. What do you expect out of that creativity piece? That's what I think the answer is research in context with focus on a system approach to risk. That's what I like, yes.
This is really just more of a comment. It was in the Nature article that you sent this morning about the lab group who, they just made it a practice.
Yes.
That every week they put everyone's name in a brown paper bag and drew out a name, and that was whose lab notebook you evaluated.
Right.
10 minutes, and you had to make sure you could make sense of
Yep.
someone else's work.
And the way that worked is we worked with them and they came up with a little checklist of how the notebook was meant to be. Can you read it? Can you read that page? Do you know what date, what equipment it came from? And then so the reviewer went the checklist, and then the checklist went back to the lab manager, and she could keep that and then see, is one person always having trouble with they can't read their lab notebooks, or never loses the source data because they don't tape it in, those sorts of issues. And as an interventionist, we had the opportunity to do that because we worked with this researcher, and before he put his grant in, I said, let's just write some QA support in here. So for your 6-month project, we can come in and get this in place. And you write that in your grant, and you can report that in your progress report that you've had an external monitor come in and check your data reconstruction. And so that's how that happened. But yeah, they're still doing that, and it's a year and a half later. Yes?
I am having so many positive, motivational reactions to this, but
That's not my usual experience.
Okay, well,
Thank you.
And I should disclose my background then,
Okay.
and my own bias in coming to this. I'm in statistics education and do a lot of helping people with their projects. And I came up through like a physics background,
Yes.
and then went to like a policy background. So I've been to different parts
Yes.
of science to see the variations and assessing, okay, what's good practice or not. And a really great statement that made sense to me by another faculty member within Ed Psych was, you can fill that in however you want, are a product of your graduate training. And so that kind of got into my head of, okay, a lot of things make sense to me, because with what I study, I'm constantly exposed to them, and I think
Yeah.
to look for them when I'm talking to other people
Yeah.
who are not as exposed to those ideas, not necessarily as a fault of their own.
Right.
Through their training.
It's so true.
So it's been this interesting case of, okay, when I have a chance to help people who haven't had the same opportunity or the same requirements, fantastic. But then sometimes I personally almost get exhausted, like can I just like take this person back and almost like ride with them on their journey when they learn this idea and that idea to like instill some of this thinking. A little more data analysis oriented, some data collection oriented. But there's this mindset, how do we make it as easy as possible by getting it in early?
Early is
so
the answer.
Right. So that's my first
A couple responses to that. One is I have a son who's a computer scientist math guy, and he hears me talking about this, and he just starts to shudder, because computer science, they document. They really have been doing what documentation way more than biologists have. And in the core group that I worked with in the Academic Health Center, one of the trainees' bosses was a computer scientist, and he was part of our panel, and he said, "We've got this, we do this." And I said, "Well, you might be the exceptional one." And we worked with his graduate student, and he was right. He had this. He had it all. He had the website, he had his procedures, he had checklists, he had documentation. He had this. But back to mentoring, or where we've been trained. I've sat in quality assurance meetings where the FDA regulators have been there, and also you hear so many times, well, you just can't get that kind of research done in academia because they just can't do it. And I just feel terrible about that, because we're missing out on really good work, right? And so I'll defend our scientists. And I have several times saying, look, we have never had training in this. No one's even had the expectation that made it really, really important that we have calibrated records. We need to integrate this into our programs. And we can do this. We can do regulatory-like or GLP-like work. There's no doubt in my mind. Yeah. Yes?
This is great. And you go into the real world.
Yeah.
Tell me from your experience when you were given this charge of what were your first steps? You have these 12 individuals?
Yes.
Yeah, I
With-
just want to know the first preliminary rudimentary steps.
Right.
Then you gain traction, and then
Yep.
I want to know your first few steps.
Okay, and so I'll tell you that, but I'll also tell you that these are the first 12, and I already am not sure I do it the same way. But the approach was, the first step was to have them do a research process. What is their research flow chart, their plan of work, okay? So for their project, let's just think of this as one person's project, or even smaller if it is one piece of their flow chart that they think is risky. And then we took that process and said, okay, now describe where the data comes and goes, and whether you feel there's any risk. So we did a risk assessment associated with those steps. So what risks do you feel are present? And it was really interesting. These trainees, they knew right away what risks were there. And so then we looked at those risks and said, okay, what kind of procedures could we put in place that would help you mitigate that risk? So if you've got sometimes the risks were, well, I've got this, there's a really good person in the lab who helps with this, but she's leaving in 2 months. How am I ever going to make sure the work is the same? Or someone really good left 2 months ago and we have no idea where that data is, how it was done. Well, that's just a walk-in to, let's get some consistent procedures in place and figure out where notebooks go. And some of it was more scientific risk, where they really, they had to use equipment that's owned by someone else. How are they going to know if it's contaminated? So we'd talk about that. So, and then after that, we said, we asked them to identify which of your procedures are routine. And then once they did that, okay, well then let's get those into, it could be an SOP, or if in your lab you use procedure steps on a wiki page, well, let's just make sure they're all there and that everybody agrees those are the procedure steps, because sometimes two people do it different. And then it was, what critical equipment do you have? If it can affect the quality of your data, if it is an important piece of equipment, let's get an ID on it and let's find the manual and let's see if you what your PI says about calibrating it once in a while. So, and we went all the way through them. We met about 7 times, and then we had the library folks come in and work with them on just what are some of the risks to data curating, to naming your columns, to looking after your data. And then we did a couple little individual. I went to their laboratories and I did a mock audit assessment to just see how they were doing, and so that's how that went. Now I'm at the point of trying to think, what would I do next? And sadly, that model isn't going to work, is it? Because I can't go into every graduate student's world as much as I'd like to. So now we're probably going to have to switch to online hybrid training courses, maybe a little boot camp that says, here's a structure, see if you can put it in place. It's a tricky one, because some of them would come, so some of the challenges they had where the PI didn't really consider this very important, so it was pretty hard for them to go in and start saying, we really should do things this way. Others, they just weren't in a position to make those changes. However, that WHO document has a wonderful table in the back that says, if you're just one person, one individual, what could you do to protect your data? And that's a good place to start. As a PI, at the very least, having a data management requirements written down that everybody is taught and that you expect them to maintain. Documentation, if you have to start anywhere, figuring out what documentation is important to you and making sure it's done well, good documentation practices, so that you can reconstruct your data. Oh, there's so much we could talk about. Go ahead. I don't want to talk too much.
So kind of jumping off from that. So I've actually worked for Carrie, so I'm very happy to see those. I didn't design the website, but...
Oh yeah, the website's, there was...
I'm happy to see the logo.
Yes.
And I'm part of a group that's actually, part of our charge is to support research in CEHD, support faculty and graduate researchers. I'm wondering, without duplicating what the university, because the university is already doing some great work around data management. duplicating
Right.
it. But as a group that's a bit closer to the subject area, a bit, we all are kind of in educational research as well. What are do you have any recommendations for things that we could do to support researchers in the college? based
Yeah.
on this?
Yeah, I think about that all the time, because that's the angle I think we need to come in at, is what can we do to support them. And it's interesting, when I met up with the library people, it was because of this little supplement grant, and it was fascinating because they talk about data management and data management plan in an entirely different way than I do with quality assurance, because they're managing data that's already been collected. So they're managing data that could be a lot of garbage data from my world. And so to me, we need both those things. So we need those folks helping us curate and keep secure and clean data. But then there's this whole piece of, what do you do to make sure your data doesn't change, and that it's source data, and that you can read it, and that it didn't get transcribed from an Excel spreadsheet. So I think the answer, and I hesitate because I'm not thrilled with it, the answer is we have to support maybe training. We have to start with training. We have to introduce this idea as a standard we should reach toward. I would love to see focus group activity where we could sit down with scientists and say, this is probably a really good idea. What would it take? Because I think about this a lot and I don't have the answers. What would it take to put it in place?
So that's been sparking in my mind a lot, because this goes back to sort of a fundamental truth, something that I learned, funny enough, in consumer behavior classes from undergrad, and that was this idea that people purchase or pursue certain things that are important to their identity. And I thought that that idea was really powerful in so many other contexts.
Yeah.
And I think the issue has come up in some of our other comments so far that changing the initial perspective is this is another barrier I
Yeah.
have to go through,
Yeah.
another box I have to check.
Yeah.
But somehow genuinely shifting that to, I want to do this because
Yeah.
I believe in it, and
Yeah.
it's part of my identity as a researcher.
Yep, exactly.
So-
And it's my data, and by
Right.
golly, I'm going to show
Right.
you. And this is the only added value other than you're going to clean up a lot of that low-hanging fruit. But the added value of confidence in your own data is really important. So what would drive scientists? Well, I kind of know the answer because we've actually had through our original Quality Central program, we have had scientists, they had to pay to get help, and several of them paid a lot, over $20,000 a year, to get a quality system integrated into their program. Now they did that because they wanted to be able to do GLP work, or one person had a big infrastructure grant and has to have all kinds of different scientists using this precious equipment, so they need a quality system in place. But others found a way to put it in their grant because they wanted to seem more critical and they wanted to be more credible. And that's truly the answer. We need the funding agencies to understand QA and to know how to review it. This is something I'd be really interested in, is reviewing the QC parts of people's grants. Wouldn't that be interesting? Because most of them just say, I have a really good CV and you can trust me, right? I mean, the ones I've seen are pretty much, I use quality control and I've been doing this for 25 years. Well, if you actually, that's probably not going to be enough. And one of the fun things is young people don't mind this a bit. For them it's helpful. And I think their expectations also are that that's how we've always done science. It's us older folks who have, done things the way we do things and just are out of time to change our stripes. And it's some who just want nothing to do with this kind of thinking. So I think we do have to start with the young ones. But boy, having a few champions of the older, successful research people who understand the value of this in terms of, okay, we're going to change, is also huge, because those are the people others will listen to.
Okay.
All right.
I want to be mindful of
Thank you
time.
so
Hey,
much.
thank you.